So today I'm going to be talking about temperature-dependent light-based regulation in the human pathogen Acinetobacter baumannii. So this is Thomas Patterson. Him and his wife were enjoying a nice trip to Egypt when Thomas suddenly became seriously ill. He was diagnosed eventually with the infection of a deadly antibiotic-resistant superbug. And that bug was Acinetobacter baumannii. So baumannii is a human bacterial pathogen that is best known to cause severe infections of meningitis and pneumonia in immunocompromised patients. The rise in antibiotic-resistant strains of this microbe has led the World Health Organization to label it as a pathogen of critical priority. And because bacteria like baumannii are no longer responding to antibiotic treatment, the search for alternative methods to treat and prevent infections has become indispensable. So beyond antibiotic resistance, what presents baumannii as a particularly dangerous microbe is its ability to respond and adapt to environments most other pathogens can't. So interestingly, in 2010, our laboratory found that one of the signals baumannii uses to respond to its environment is light, and that this response is also dependent on temperature. So the ubiquitous nature of light in the natural environment, skin infections, or even for therapeutic purposes makes the response to it as a stressor extremely prevalent. So up to date, most findings of light-mediated regulation have been surrounding 24 degrees Celsius, or about room temperature, specifically with the photoreceptor BLSA. So BLSA has been shown to regulate a myriad of virulence factors in a light-dependent manner, including motility, iron uptake, and biofilm formation, and more. However, the full sphere of influence, as well as the mechanisms through which BLSA functions, remain unknown. So through the support of the Beckman funds, we were able to not only expand BLSA's influence to include baumannii's response to oxidative stress, but also further determine the importance of C-terminal residues of BLSA and its regulatory mechanisms. So first, oxidative stress is faced by pathogens both in and outside of a host in multiple manners. And pathogens respond to this stress primarily with the antioxidant enzymes catalase and superoxide dismutase. Specifically, many multi-drug resistant and virulent strains of baumannii have been shown to have significantly higher expression of these enzymes compared to their avirulent counterparts. So we were able to show that BLSA allows fine-tuning regulation of both of these enzymes based on light. Interestingly, while catalase activity is upregulated in the light in a BLSA-dependent manner, SOD levels are upregulated in the dark in a BLSA-dependent manner, pointing to the likelihood of two different sources of regulation. So we were also able to further elucidate the mechanisms through which BLSA could regulate. Although it has been shown that BLSA interacts with protein partners to control some of its regulon, the structural components of BLSA itself that allow these interactions and the potential other protein partners and regulation manners have yet to be understood. So our laboratory had previously shown that while BLSA's N-terminus is imperative for the light sensing activity, its C-terminus is what allows its regulatory functions. So we've studied the importance of certain amino acid residues in BLSA's C-terminus and the light-mediated regulation of motility. So today I will be focusing on the most C-terminus residues, K144 and 145, as well as the last 5 most C-terminal residues, which I'll be referring to as delta 5 from now on. So we were able to extend the characterization of these particular residues for both catalase and superoxide dismutase activities as well. So it's also really important to note that only very recently, a study on the structural biology of BLSA has revealed its interaction with the protein partners and transcriptional regulators FIR and BFMR. However, it's also important to note that there has been little to no focus on the pathophysiological implications of said interactions, and interestingly, the most C-terminal alpha helix, or the region on which we focus our studies, was unable to be resolved. It's also important to note that fur has been shown to regulate SOD levels in A. baumannii, while BFMR has been shown to regulate both motility and catalase activities. Notably, the same catalase homolog that we showed is regulated by BLSA. So when finally looking closer into these actual amino acid residues, we were able to show that one lysine residue was shown to be important in light-mediated regulation of both catalase as well as motility. But interestingly, the last 5 amino acid residues, or the delta 5, although showing absolutely no importance in motility, were essential for the regulation of both SOD and catalase activity levels. Most notably, the delta 5 was characterized by an overall decrease in catalase activity despite a maintenance of the light-mediated regulation comparable to wild type. Therefore, the complete role of these residues in the BLSA regulation, both dependent and independent of light, is far more complex than originally assumed. Overall, from these data, we were able to decipher a putative model of the regulation of SOD and catalase activities by BLSA, which I can go into more detail with in my poster, as well as the importance of the amino acid residues in general regulation. So by comparing the importance of these amino acid residues in motility, as well as catalase and SOD activities, we are able to clarify the mechanisms through which BLSA might regulate cellular activities. Specifically of interest, we were able to highlight the fact that BLSA uses different amino acid residues to interact with its different protein partners, indicating the specific nature of these bindings, as well as the advanced functioning and specificity of BLSA. So as alluded to before, A. baumannii light-mediated regulation is also temperature dependent. Specifically, BLSA is not considered functional at 37 degrees Celsius due to insignificant expression, as well as hindered light reception abilities due to conformational changes. So our laboratory had previously shown a BLSA independent light-mediated regulation of biofilm formation at 37 degrees Celsius. But until now, no efforts at looking at global light regulation at 37 degrees Celsius has been attempted. So through RNA-seq analysis, we were able to find a host of functional groups of genes that are being regulated by light at 37 degrees Celsius, most of them particularly being involved in metabolism. But of particular interest, we have found that at 37 degrees Celsius, the genes encoding for the transfer of a multi-drug resistant conjugative plasmid were being upregulated in the dark. And so we were able to confirm in vivo that conjugation was indeed of this plasmid higher in the dark. So importantly, this plasmid encodes for antibiotic resistance to multiple drugs, and preliminary data have also shown that this plasmid mediates many important virulence factors in A. baumannii. So ultimately, these studies have been able to show, again, how A. baumannii responds to environmental signals, specifically light, and how it is able to take this signal and confer some kind of response through regulatory mechanisms. So ultimately, understanding how A. baumannii responds to environmental signals is imperative for the development of alternative methods of treatment and prevention of infections that, as in the case of Thomas Patterson, would otherwise seem unstoppable. So with that, I just want to say thank you to Miami for providing fantastic faculty, and specifically to all my advisors, specifically Dr. Actis, as well as the Beckman Foundation and NIH for very important funding. And then thank you to you for listening to this talk. Hope you have a wonderful day.